Okay, so we're going to go ahead and get restarted. Jack Beauchamp's going to present next, and he's working on reagents based on a proton transfer chemistry or free radical chemistry to address the, and I would say, very rich challenges of glycan structure.
Thank you. I want to make sure I give credit here to Jinshan Gao, who's the one that's almost entirely responsible for the work that we have done with determining the structures of glycans. But it's based on a lot of other studies in the lab done by Chang Ho San and Daniel Thomas in the group. So I've added their names here to make sure they get the credit for this up front there. So this is part of a program we've had over the last 5 or 6 years to see if we can take the information that we've learned over the last 40 years about the properties and reactions of ions in the gas phase and free radicals and harness this to derive and use new reagents that can be applied to problems in proteomics and glycomics. One of these efforts involved the assembly of an isobaric tag. We've developed a novel chemical cross-linker. I'm not going to talk about these today. And then most recently, the glycan sequencing, which is largely based on work we did earlier with free radical-initiated peptide sequencing reagents, or FRIPS reagents, as we call them there. The proteomics reagents here, the Caltech isobaric tag. We heard earlier this morning that it would be nice to be able to do isobaric tagging. That didn't cost a fortune for each experiment. This particular isobaric tag we're giving away to people free on request, and it costs actually, if you synthesize it yourself, about 2 to 5% of what the iTrack and other reagents actually cost for an experiment. So you probably could do a large amount of material. But we have a lot of this that we made, and we give it out to people. It's patented, and we figured if enough people find it useful, it'll enhance the opportunities to license it. We have a clickable cross-linker, which is cell permeable, so you can actually cross-link in vivo. The usual reaction centers here that will couple to a lysine, to a primary amine. But we have a tertiary nitrogen here, which serves a couple of functions. It's protonated and as a result makes this cationic, and that enhances the cell permeability. We have a bioorthogonal function here so that once we isolate the cross-linked tryptic peptides, we can click on biotin for the isolation. And it's also designed then when you do collisional activation, it releases the biotin so it doesn't mess up the mass spectrum, and the biotin itself serves as a marker for the fact that the peak that you've selected at that point is a cross-linked peptide. So you ignore it if you don't see the marker there. So it's very versatile. Again, if you want some to try it, we've been giving this to quite a few people here and in Europe. And the free radical-initiated peptide sequencing, or FRIPS reagent, I will explain a little more on the following slides. But the reagents for glycomics, and we'll discuss these in great detail, are really based on the work that we did earlier, mainly with the FRIPS reagent. So I'll tell you basically what these are and how they're used for glycan structure determination. The initial part of this work just appeared a few months ago in JACS. So the free radical-initiated peptide sequencing, this started in 2005. We had the idea that we could couple a reagent to a functional group and a peptide, picking for generality, the N-terminus, and then have the ability to generate a free radical that could in turn be used to selectively cleave peptide bonds. And the reagent we have is actually quite simple, using a tempo leaving group. So this bond is very weak, and when this is coupled to a peptide, collisional activation, you lose the tempo, the stable tempo radical, leaving behind an acyl radical that then attacks various sites on the peptide and gives you sequence information. Now, it's the same idea that I'm going to show you works well with glycans there. So this is, shows the Frips derivatized tryptic peptide that's indicated with this sequence. And when you do the tempo Frips experiment, this indicates the fragmentation processes that you see. It's all radical ion chemistry, but like the UV photodissociation, you get much more sequence coverage. In this case, 91% sequence coverage compared to 55% here with CID. Now, this is cheating a little bit because we've got, of course, protonated arginine here, but there's a couple of aspartic acids, and we know salt bridge chemistry tends to give the majority of the cleavage at those two sites. So this may not be entirely a fair comparison. But the free radical sequencing works quite well. And the major series of ions that we see is the result of a hydrogen atom abstraction, actually from a beta hydrogen or from an alpha hydrogen in the case of glycine, followed by a beta cleavage. So in this case, it gives you ANX type ions depending on which side the charge is actually retained on. So it's hydrogen abstraction followed by beta CC cleavage that's responsible for the fragmentation there. Side chain losses here, you can distinguish leucine, isoleucine easily. You don't lose PTMs in that, so you can still identify phosphorylation sites, so the same efficacy that you can with ECD, ETD. You can generate actually peptide products that have a well-defined radical center and then examine its subsequent chemistry. And disulfide bonds are cleaved in preference to backbone cleavage. So you can actually map disulfide linkages in proteins. And if you're interested in this, just let me know and I can send you a preprint that's available. Now the question is, how can we develop a similar reagent to analyze glycan structures? And in biological systems, redox chemistry, free radical chemistry is known to be responsible for the decomposition of carbohydrates along with acid-base chemistry there. Well, glycans, glycomics, this is actually a cartoon that Steve Carr showed if you heard his lecture last night, where you have the gene is of course defining proteins that are expressed. They get post-translationally modified. They assemble into protein complexes. But I've added on here a couple of Christmas trees to indicate they weren't shown last night, that one also has glycosylation that is extremely important in all aspects of cellular functions, including cell surface glycans. Now, they're not templated, right? So you don't have the wonderful connection between the protein structure and the gene sequence here. They're generated instead by a series of enzymatic reactions, and it's very easy for these enzymatic reactions to get it wrong. So you occasionally have not one structure, but a distribution of structures of glycans that you're dealing with. And basically, what we'd like to do, the simplest thing would be to cleave the glycosidic bond between the subunits here. Now, one other feature of glycans that makes them particularly challenging is that unlike nucleic acids and proteins, which are linear and which have a limited alphabet, so, you know, DNA, you've got 4 bases, proteins, you've got 20 common amino acids, so you know what you're dealing with. Now, there are, of course, PTMs that modify some of these that make it a bit more difficult, but the sugars really get you. There are a lot of different subunits that have varying substitution in terms of functionalities that are added on. And you have several sites at which the actual branching may occur. So you get these highly branched structures, not linear at all. They look like trees there. In fact, I was skiing over President's Day up at Mammoth, and I stopped, and my wife says, "Why are you photographing that tree?" And I said, "Well, it makes me think of a project back in the lab there." And so we're dealing with trees here, or at least with a well-defined bush. Now, the simplest type of reagent one might think of developing to mimic nature here in constructing a reagent would be one that simply does acid-base chemistry on the glycosidic bond. And usually enzymes arrange a couple of acidic residues. You can use other residues here as acids and bases as well. But such that you have a proton acceptor and the ability here then to activate a water molecule that displaces the OR group, which in this case is cleavage of the glycosidic bond there. And the reagent that actually does that, very simple, easy to synthesize, uses a functionality here that can specifically react with a carbonyl, so it will add to the open form of the reducing end of the glycan, and you attach an acidic functionality to it. Now, this is very carefully picked to have a proton affinity that is in what we call a Goldilocks zone. You don't want it too high, you don't want it too low, because you want the charge to stay on the on the reducing terminus of the glycan. At the same time, you want it to be available to use as an acid. You don't want it to kind of hug up too much to the proton. So pyridine works extremely well. We have, in addition, then, a reagent where we can generate a free radical to do the Frips type chemistry. And we call this free radical activated glycan sequencing 1 or frags 1 here. Now this one has a methyl, so we get rid of the proton, so we won't have the acid-base chemistry. Instead we'll have just the free radical chemistry, and then frags 2, if you want the whole enchilada, basically you've got the proton and the free radical. So it generates the most complex spectra, but also the greatest amount of structural information in a single experiment there. And then finally, in the real world here, we have a resin-supported reagent that is equivalent to the frags 2, a slightly different functionality here for coupling to the glycan there. So how does this work? We take our reagents, we couple them to the glycan, and in this case, we're just taking maltoheptose as an example. And then isolated in the mass spec, singly charged, and collisional activation then does the rest. So you don't need laser, ECD, ETD, any other attachment. You can do this in any type of mass spectrometer where you can do CID there. The nomenclature here is the standard Costello nomenclature. For the glycosidic bond, we're going to be looking actually at the formation of Y-type ions, because remember the charge is going to stay on the reducing end of the oligosaccharide, and then to get more complete structural information, though, we're going to need cross-ring cleavages there. So this is the very simple spectrum you get from the proton reagent. You get peaks here. It's just a series of Ys that give you basically cleavage at each of the glycosidic bonds. You notice the Y6 here, way far away from your reagent, is the most abundant, and we think that has to do with the preferred conformation of these things in the gas phase there. But it's quite different from the CID, because if you start with either protonated or sodiated maltoheptose, you get not only Y ions, but you get A, B, C, X, and Z ions there. But the charge can stay on either end of the molecule when it fragments, so the spectra are very complex there. And the chemistry here is just acid-base chemistry. We're not sure as to what the exact steps are, but we imagine that the proton is handed over to the oxygen. That makes it possible to then cleave the glycosidic bond, and then this acidic species gives the proton back to the pyridine, and you're ready to go to the races again. So in this case, you retain the capability to do further cleavage reactions, and I'll show you how that's useful for large glycans there. This is actually just MD simulation. This is one of the low-energy structures of the glycan, the derivatized maltoheptose, and it folds back on itself to maximize hydrogen bonding, and we think this is what promotes reaction at the opposite end of the oligosaccharide there. Now, we It skipped one there. So this is the now the frags 1. So this is the free radical reagent. We blocked the acid-base chemistry, and now we're getting a lot of cross-ring cleavages, which is what you need to find out where the different functionalities and the branching points are for the branched glycans there. The mechanism here involves, as it does with the FRIPS, the abstraction of a hydrogen atom from the ring, and all of the CH bonds here on the ring are about the same, around 94 kcals per mole based on calculations, DFT calculations, and we actually see reaction at every site on the ring there. And once you abstract a hydrogen, you have a pair of beta cleavages, and that allows the cross-ring fragmentation to produce useful structural information there. So this summarizes basically the prags. We get acid-base chemistry, we get just free radical chemistry. The combined reagent, the frags 2, spectra more complex, but all of the cleavages are present. Now computers can of course sort this out, but if you're grad students, it's easier to look at these there. Now, branched glycans, or these bushes, are what really present a challenge there. And this is lactoin difucohex, let's just call it LNDFH2 there. And it exists as a couple of isomers. This happens to be one of them. And this would be the symbolic representation for the glycan. And let's see what happens now when we do the CID. So this is mainly glycosidic bond cleavages, right? But it's not a simple pattern the way the linear saccharides give a simple pattern there. It's much more complex, but you can still actually analyze this in terms of it's mainly glycosidic bond cleavages that are occurred. And more important, now since with the protonation reagent, that proton always stays on the reducing end of the carbohydrate. So you can do this over and over again, right? So you can clip off just one sugar, and then you can isolate and do CID on that particular species and look at all of the fragments that result from that. And this is actually the result of three steps of MS-MS on the system. And we call this a deconstruction diagram or a decon diagram because from these data you can actually go back and piece together that this is the arrangement of the subunits in the glycan. All right, so this is it's the simplest reagent, but it's the one that's our go-to reagent to initially characterize an unknown structure there. Now, the free radical chemistry of these, both the frags 1 and frags 2, is a lot more complicated there. And we compared two different isomers here, the LNDFH1 and 2, and you notice they differ. I've highlighted it. They differ in the location of a fucose subunit there. So if we look just at the difference in the proton reagent here, it's already quite different. All right, so you get a lot of information that it's not totally unambiguous as to the structure. You have to read the paper to see that in there, but you can tell, in fact, that they're different structures there. And when you do the frags fragmentation here, they're very different, and that's what actually gives, with the cross-ring cleavages, gives you the complete structure. This shows the frags 2, which is both the acid base, and it's only a subset of the total number of fragmentation processes there. But basically, it's the systematic and predictable X plus Y and Y plus Y ions here that nail down this structure. Now, fortunately, computers can sort this all out. But the important thing is that you can easily distinguish these two structures. I should say not just distinguish them, but actually characterize each structure separately there. Now, the real world, of course, is not as simple as buying a sample from Sigma. So the first example that we looked at is one that people use to kind of test the methods for analyzing glycans there. It's RNase B, and this is a substrate that has a site of glycosylation where there's several glycoforms attached to that position, and it's cheap there. And we cut the glycans off using PNGase F, right, for the in-linked glycans, releases them. For the ribonuclease B, known glycan structures, I put that in quotation marks because a lot of papers we've looked at now where we're looking at glycan structures, let me tell you something, they're not right. I should say, if you read the fine print, they'll say that this is our best guess for the structure based on a lot of known structures, all right? And I'll show you an example of that there. But the glycoforms here are the main ones are the 7, 8, and 9-mer here. So now to use this in a practical sense, we've got our mousetrap here that can shred attach to and shred the glycans, but we need to be able to separate them out from a complex matrix, especially if you do cellular level glycomics there. And the way we do that is to use a disulfide linkage and then couple it through an additional functionality here to resin bead. And we use a reagent that will specifically condense with an aldehyde or a ketone. So again, it reacts with the reducing terminus of the sugar. And the particular one we picked here was the combination of the proton and the free radical reagents there to get the most complete coverage. And then when you're done with the cleanup, you can use DTT to cut off the resin and do the mass spec analysis there. So that just shows the experimental protocol that I've just described here for dealing with a sample with a proteoglycan that you say have removed from a gel there. And the one that we tried it on initially here was this RNase B. And they're not quite showing here, but that's just as well. A couple of previous studies had been done using mass spectrometry recently to characterize the 7, 8, and 9-mer there. And these two studies actually didn't completely agree as to what the structures were, which was one reason we picked this as an interesting problem. Researcher 1 had this structure, researcher 2 had this, similarly here and similarly here. All right, so we checked this out with our reagent, and they got that right, and that's wrong. For the 8-mer, that's right, and that's right. Both structures are actually present, so it's a mixed structure. For the 9-mer, that's wrong, and that's wrong. They're both wrong. And in fact, the two possibilities that one has for the 9-mer, I'm not going to show you all these data, are this, okay? That after the first branch, there are 4 of the nano subunits on one of the 2 branches. We can't actually distinguish this one from this one, unfortunately, but we know it's neither of those. Yeah. So this would be, they may actually both be present there. But this would, I think, require further characterization, perhaps with ion mobility spectrometry, to be able to distinguish those in comparison. The fragmentation patterns, if you had the bona fide materials, could also be used then to distinguish them, because they are different, but they don't really distinguish those two possibilities there. So that's where we are. If you want some of this reagent, we are giving it away. All right. I had Jinshan actually make quite a bit of it. He's now teaching at Montclair State University. Has a teaching job there in the chemistry department. But he will also provide assistance in using these reagents there. So in conclusion here, we've got these biomimetic reagents there, the proton reagent. And let me emphasize with the proton reagent, this ability to do the MS to the nth is really quite useful. you can think of it as a reagent that you can use over and over again. The free radical reagents are like a rattlesnake. It's got one good bite, but now your radical's off somewhere in the molecule, and you can do MSMS in there, but it doesn't always then reference everything back to the back to the reducing end of the sugar there. And the resin-supported analog there give you capability for glycan structure analysis. There's several groups at Caltech actually that are using this and quite excited by what they're getting out of it there. So we've got acid-base chemistry. We've got the deconstruction diagram there that uses MS to the nth data to get more complete structure. The cross-ring cleavages here due to the free radical chemistry are what actually gives you the position of substitution in the and also the position of modifications such as sulfonation of the glycan there. Now, it would be very easy to modify these reagents to incorporate addition of an affinity or capture reagent, such as we did with the resin-supported unit. But we could also use an alkyne attached to the reagent and then click chemistry to attach biotin that could then be released as a marker for doing the further structure analysis of the attached glycan there. And you could also actually do comparative glycomics by incorporating an isobaric tag such that when you release to the marker ion, it separates. Initially, the whole system has the same mass, but as the two halves separate, you got a little deuterium on one and not on the other, and vice versa. Now you get a mass tag that you could use for protein, for glycan quantification. And that would be very useful, actually, with respect to comparative glycation states there. So the people here that did this work, Jinshan Gao, did all this work in 9 months. Amazing guy, just an amazing guy there. Initially got a lot of help from Daniel Thomas and Chang-Ho Sun, who had developed the FRIPS chemistry and refined it so that it is more or less automatic there. And then various sources of funding that have helped with our financing our work. So thank you.
We certainly have time for questions.
Yeah. Have you tried like the home run experiment of doing this on a glycopeptide and not releasing the glycan and seeing if you get the effect?
We've tried it on a glycopeptide. I forget the particular one. So we just used the FRIPS reagent there. The FRIPS reagent tends to react mainly with the peptide portion and not the glycan. So the activation energies for abstraction of hydrogen from several of the residues that commonly appear, usually it's things like phenylalanine, where you've got a CH bond there that's relatively weak. So we saw mostly the peptide fragmentation, but if we'd seen actually competitive fragmentation, that would have been nice. But you can tell where the site of glycosylation is. We're still trying to figure out a way to do that. Yeah, and so you get the combined information. Well, with the FRIPS reagent for the tryptic peptides, we've looked at a lot of phosphorylated peptides, and the answer is yeah, you can tell where the site of phosphorylation is, because it's not. You mean sites of phosphorylation on the glycan? We haven't looked at that particular modification in there. We have trouble with GAGs that are highly sulfonated. We still see a lot of loss of SO3 when we do the collisional activation, not entirely. So we can get a lot of information about them, but we're trying to figure out ways that we can actually, we're working on an anionic reagent, right, which does the same kinds of things. But we don't think it's going to have the problems associated with loss of the SO3 from the sulfonates there. But we haven't looked at any phosphate substituted glycans.
The Fritz reagent with the glycan, you sort of talked about as a snake bite, one good bite.
Yeah.
Is that controlled by the conformation of the glycan, or is there some?
No, it's just that once you get the initial hydrogen add, the question is, why is it just a snake bite? Oh.
Is the selectivity, do you have a chance to have selectivity, or is the selectivity dictated by the conformation of the?
There is some selectivity. We generally see reaction everywhere to some extent, but some of them are much more prevalent. Some cleavages are much more prevalent than others, you know, maybe a range of 20 to 1 sometimes. And that probably has to do with conformation there. Even with the maltoheptose, in that the cleavage of the Y1 ion actually doesn't fit the overall pattern. But in fact, there's a conformation that leaves the reagent almost ideally suited to just reach out and grab that, transfer the proton over to that particular glycosidic bond there. But when it folds up, then you get more reaction at the end of the glycan.